Okay, welcome everyone. Thank you for being here. I'm going to get started really quickly because I know Dr. Milewicz has a lot of exciting information to share with us, and we have only the hour. We can't force her to stay longer today because she has lots of work to attend to, so we are going to be efficient. But I will just quickly do an intro here. And we all know where we're at. So my name is Ellen Hostetler. I'm the Chief Science Officer for the John Ritter Foundation for Aortic Health, and we are very pleased to be hosting this event, the third, I think, in the ACTA2 series that we're doing with the Genetic Aortic Disorders Association Canada. And I'm joined by Chrisanne Campos here today, who's their scientific director. And I won't read through all this because probably you know who we are if this is the third one you've been with us, but please always feel free to reach out to us if you need information. We're on social media. You can send me an email. As always, we're pleased to have Dr. Milewicz as our longtime partner. She serves on our board and also on our professional advisory board. So excited, especially to have her as a presenter again today. And Chrisanne, I'll let you take it from here.
Yes, thanks, Ellen. So yeah, again, as Ellen said, I'm Chrisanne from GADA Canada. GADA is a registered charity patient advocacy organization in Canada. And we've been supporting research for genetic aortic disorders for the last 23 years with a generous support from the Timothy Foundation. So any information you'd like to learn about GADA's funded grants, please visit the GADA website. And more importantly, what I'd like to point out here is our support for the Montalcino Aortic Consortium research that is led by our invited speaker here, Dr. Milewicz, who is leading the registry at the University of Texas in Houston. Any questions to register for the MAC research or to be enrolled in MAC research, please reach out to the MAC program at the UTHealth or email me or Ellen for more information about MAC. Before I can introduce the speaker, who everyone already knows, I'd just like to quickly go over the disclaimer for this talk, that this information that's going to be presented in this webinar is for educational purposes only and shouldn't be, any information shouldn't be undertaken without consulting your doctor or your specialist who's following your case. And if you have any questions, please feel free to use the little box on the bottom. You can use the chat box or the Q&A box. As Dr. Milewicz goes through her talk, you can put in your questions anytime through the talk. We're happy to address those questions either during the talk or after the talk. So I'd like to quickly introduce our one and only speaker, Dr. Milewicz, the specialist in ACTA2. She is the President George H.W. Bush Chair of Cardiovascular Medicine, the Director of Medical Genetics, and the Vice Chair of Internal Medicine at the University of Texas in Houston. Her work includes translational studies focused on genetic predisposition to aortic disease. She and her research team first identified ACTA2 as a cause of genetic aortic disease in 2007. And she is going to be talking to us today about ACTA2 molecular research today. And so without further ado, I'd hand this over to Dr. Milewicz to take over.
Hi, everybody. Thank you for that nice introduction, Chrisanne. And she's right. I'm going to talk to you today about our studies on how defects in ACTA2 cause all these different problems with the arteries. So let me see if I can get my slides. Okay, everybody can see my slides okay?
Yeah.
Good. So when I gave a talk, about a month or 2 months ago on how ACTA2 variants, are what we also call mutations in the ACTA2 gene, cause thoracic aortic disease, but also cause early onset coronary artery disease due to atherosclerosis and early onset cerebral vascular disease in a moyamoya-like pattern. And I didn't have time during that talk to really talk about the molecular biology. And quite a few people wrote into the John Ritter Foundation and GADA and said they really wanted to have some idea in terms of what's going on at the molecular basis. So I'm going to talk about that and try to make it very simple enough that you don't have to have a PhD in biology or molecular genetics to understand this. So just to start out from the beginning, as I mentioned in my previous talk, that I've been very interested in identifying the changes in our DNA that predispose people to aortic aneurysms, because aortic aneurysms predispose to life-threatening acute aortic dissections. And if we know who's at risk, we can go in and fix the aneurysm and completely prevent those deadly aortic dissections. And so I chose as sort of the focus of my career is to identify all the genes that predispose to these diseases. And so we've been working many years trying to identify these genes. And here's a list of genes we have, my lab, along with some other labs around the world, have identified for this disease. And so these are all genes that cause aortic aneurysms and dissections. And I'm going to change my pointer so that it's a laser pointer. And this is telling us something about the disease. And I talked a little bit about this before, that it's telling us that that the smooth muscle cells, which are the cells that are in the aortic wall, that these mutations disrupt the ability of that smooth muscle cell to contract. So the smooth muscle cells surround every artery in your body, and I'll show you some pictures of that in just a minute. And they have to contract down every time a pulse of blood goes through the artery. And they contract just like your muscles contract or your heart contract. It's a muscle cell. And its trigger to contract is the pulsatile blood flow. So as you're sitting there and blood's flowing through your heart, flowing through your arteries, because your heart is pumping, hopefully, that with every single pulse pressure from that beating heart, your smooth muscle cells are contracting. And that what happens with all these mutant genes is that the smooth muscle cell doesn't contract as well. And that ultimately leads to aortic aneurysms and dissections. And I think there was a talk last week on how we're trying to figure out this link to a problem with the smooth muscle cell contracting and aortic disease, and specifically to dissection. We have no drugs that prevent dissections. And that's actually a part of another research program that I have going on to find those triggers for aneurysms, but specifically find the triggers for dissection, because that's what is life-threatening. And like I said, we don't have any drugs for that. And then, as I talked about previously, there is one gene that we got very interested in because it caused not only thoracic aortic aneurysms and dissections, but also caused early onset coronary artery disease due to atherosclerosis. And also for the arteries in the brain to be blocked in a pattern that's consistent with a condition called moyamoya disease. And I'll go through that and just briefly in the next few slides. So here's the family that we use to map and identify ACTA2 as a cause of thoracic aortic disease. This is a pedigree so that men are squares and women are circles. No surprise there. And somebody that's married has a line, people that are married have a line between them, and then the line down goes to their children. So this couple here had 3 boys and two girls, and then they went on to have children, and there's another generation down here. So this is the family, and everybody in black has those thoracic aneurysms predisposing to dissection. And this is actually the family that we use to identify the ACTA2 as a cause of thoracic aortic disease. And this family has this arginine, a change in the ACTA2 gene such that it changes the corresponding protein, which is called smooth muscle alpha actin. It changes one amino acid in that protein from an arginine to a cysteine. So this is the variant in the ACTA2 gene that's causing aortic aneurysms and dissections in this family. Now, everybody in black does not carry the variant because you have one normal gene, one mutant gene, 50-50 chance of passing it on to the next generation. So, but you can see there's a lot of people here that have the mutant gene but don't have aortic disease. And what we were able to figure out was that instead of having aortic disease, these people in these families instead had early onset atherosclerosis with no risk factors for atherosclerosis. And if we put the people with atherosclerosis in this pedigree as early onset CAD in red, you can see once again that people with the orange around them, that if people in the family that carry the variant, that they can have either thoracic aortic disease or early onset CAD. We now know that some of these people have both diseases. So even if you have atherosclerosis, we still have to watch you for thoracic aortic disease and vice versa if you have this arginine 149C variant. And this was early onset coronary artery disease. So coronary artery disease is what caused heart attacks. And this individual had his first heart attack at the age of 33 and went on to have a second heart attack at a few years later. He had no risk factors for coronary artery disease. And most of you probably know that coronary artery disease does not start typically until people are in their 50s and 60s. But in these families, it was starting even very much younger than that. And we also found out that people with different variants in ACTA2 did not have coronary artery disease, but instead had a disease called moyamoya. And that's where the arteries that go up into the brain, called the carotid arteries, are wide open. And then when they go into the brain, they suddenly get blocked in these people. But it's not atherosclerosis. If you look at the pathology, which is here, it's not typical of atherosclerosis. And this was caused because the arteries are getting blocked, these people were having strokes. And once again, these strokes can start very young. This young lady, and this is her imaging up here, where her arteries are the white stuff is her arteries coming up into her brain, those carotid arteries. And then over here, you can see it starts suddenly. And it's because there's a blockage there in the artery. And over here, you can see it goes from being pretty wide to being very narrow and almost block. And it's not surprising given the fact that these are two of the major arteries that provide blood to the brain, that she actually ended up having a stroke at the age of 16 because of this blockage. And when we looked at families with this variants that disrupt a different arginine or R, this is 258 instead of 149, they were having the cerebral vascular problems, but this is not atherosclerosis, and they weren't having coronary artery disease. And after we published this data, we went on to show that we found that there were children that had both thoracic aortic disease and this early onset moyamoya disease, the cerebral vascular disease, at very young ages. And this was, and along with that, they had problems with their eyes and their gut, and and their bladder failing to contract properly, and also lung problems. And all these children have the same change in the ACTA2 that disrupts not arginine 258 or arginine 149, but instead arginine 179. And it shows up new in them. Neither parent have it. And it's what we call a de novo variant or a new variant in the child. And it's nothing the parents do. It just shows up by chance. So what are these smooth muscle cells, and what is ACTA2 in the smooth muscle cell? So an artery actually has 3 layers. The inner layer is called the intima. The middle layer is filled with smooth muscle cells. So that's the muscular layer that contracts with every pulse pressure. And then there's an outer layer called the adventitia. But you can see this muscular layer is the thickest layer of the wall. And what happens with both atherosclerosis and moyamoya disease is that the arteries that are the smooth muscle cells that are in the wall actually end up migrating and going into the inside of the artery. And this is called the formation of a neointima, a new intimal layer. And the cells end up moving and they start crawling along, and they move from the wall, where here in a normal artery, they're staying in the wall. But in a disease, an ACTA2 patient with either the 149, 258, or the 179, their smooth muscles are tending to leave the wall of the artery and move inside of where the blood flows, which we call the lumen, the inside of the artery. And with that, they end up starting to occlude or totally occlude the blood flow that's supposed to flow like this. But with atherosclerosis, there's typically a lot of cholesterol deposits and inflammatory cells. But in the moyamoya disease, it's almost pure smooth muscle cells that are migrating into the artery and blocking the artery. So we started out from the very beginning with a hypothesis that with ACTA2 mutations, there's 2 different things that can happen with the arteries, a few different things that can happen with those smooth muscle cells. And for all the variants in ACTA2, there is a decreased contraction of the smooth muscle cells. And in the large artery with lots of pressure from that pulsatile blood, that leads to thoracic aneurysms and dissections. But with a few variants, and specifically 149, 258, and 179, that for some reason they cause the smooth muscles to migrate and potentially proliferate more. And in the small arteries, that ends up closing off the arteries, leading to coronary artery disease and moyamoya disease. So this was our initial hypothesis. Obviously, these are 2 different processes, so we needed to separate that out into studying these different mutations. And when the first question we asked was what was different about these mutations that cause the smooth muscle cells to start moving into the inside of the artery and instead of staying in the wall where they're supposed to stay. And here's all the variants that we have in ACTA2 to date. And here's just a 3D reconstruction of the protein and where they are. This didn't really tell us too much about where the mutations are. Like 149's over here and 258's up here, but it doesn't really tell us that much. There's a lot of variants that don't disrupt that R or for arginine, and they're all down here, and they just, right as far as we know, just cause thoracic aortic disease. But if we look at all the arginines, we're still trying to figure it out, but we know this other arginine can cause coronary artery disease along with that 149C. And then we have these 2 green variants that cause the moyamoya-like disease, and that's 179 and 258. And I'll get back to some more details about some of the newer variants that we've found that kind of overlap with 149 and 258 in a minute. But first, I want to just really start out by talking about the 149 and how we figured out what the pathway was to atherosclerosis. So I think everybody knows that high cholesterol is in your blood is the major risk factor for atherosclerosis, and that if you take a statin, or that statin blocks cholesterol synthesis in the liver, and that pulls the cholesterol out of the plasma and lowers the plasma. And statin has probably been the #1 drug in terms of saving lives worldwide. Saving lives of people that are at risk for atherosclerosis. Atherosclerosis is still the #1 killer worldwide. And with using statins to bring down blood cholesterol levels, it's really helped to do away with a lot of atherosclerosis. So the first thing we did was we engineered a mouse to have that arginine 149C variant, and then fed it, put it on a background of an ApoE mouse that gets atherosclerosis, and then asked, does the ACTA2 mouse have more atherosclerosis? That when we put in that ACTA2 mutation, does it have more atherosclerosis than your typical atherosclerotic mouse? And here is the males and females with that ApoE background, because for mice, we have to do something to them to get them even to form atherosclerosis. So in this case, we put them on this, we modified their genetics a little bit. And what's shown here is this is the aorta coming up and going down all the way down to where it branches into our legs. And these are the femoral arteries down, the iliac arteries down here. And all the red parts, this is the stain we use that stains that cholesterol, so we can pick up the plaques with cholesterol. And you can see that most of it is up here in the first part of the aorta. In this case, we've taken the aorta and actually just opened up the entire aorta and sort of split it open so we could see from the inside looking for those plaques. So that's why there's two parts up here. But then if you look down here, you can see right away, without even like having to quantify the amount of red, that there's a lot more atherosclerosis when we engineer in that ACTA2 arginine 149C variant. So this really reproduces what we see in patients. And we went on to show the atherosclerosis formed faster, it was formed quicker in every single part of the aorta. And it also went up and formed atherosclerosis in those carotid arteries that go up to the brain. And this is just all quantitation over here. And this just shows you we could take the ACTA2. Remember, mice just don't get atherosclerosis. And we could take the arginine, the ACTA2 arginine 149C mouse by itself without the ApoE and so on, and it would form atherosclerosis even without the ApoE background. And I don't think that's actually been seen in a mouse model previously. So now we know that this mouse really shows what goes on in the humans. And so we wanted to figure out what the pathway was from this. And I want to say, if you look at the blood cholesterol between the ApoE and the ACTA2 arginine 149C, there's no change in blood cholesterol. Both of them have slightly elevated cholesterol, but there's no difference. So it's not due to the blood cholesterol. So we wanted to see if we could figure out, we've got this mutation in ACTA2, which actually encodes a contractile protein in the smooth muscle cells, should have nothing to do with atherosclerosis, and figure out how it's causing this disease. So the first thing we found out was that the ACTA2 arginine 149C change cause the protein to not be folded correctly. So the ACTA2 makes this protein called smooth muscle alpha actin, and inside the smooth muscle cells, it forms long filaments, and that's the filaments there that allow the smooth muscle cell to contract, because those filaments are critical for contraction. And so what you want to do is the protein's being made, you put it into a complex so it folds properly before it starts polymerizing, because otherwise it would mess up the polymerization. And so what we found was that normally the actin goes into a complex called the CCT, and it looks like a little pot, and that goes in, it closes, folds it, and then lets it go, opens back up, and that's the way it functions. And what happened with the 149C was the actin monomer would go in the pot, and it would close, and then it wouldn't open back up and release the protein. And so we knew early on that there was problems with the folding of the protein in this complex. So we also knew that with atherosclerosis, that the smooth muscle cells, those cells in the wall of the artery, they tend to proliferate out of the wall. This is with, in any mouse model or human with the disease, the smooth muscle cells come out of the wall, and they can start taking on the characteristics of other cell types that we know are there in the plaque. And so we really think that so we wanted to see if our smooth muscle cells were changing phenotype more rapidly than wild type cells. And I think I'm going to skip some of this, but what we found was we could assay for those changes in phenotype that occurred back here with these changes here. And so with these changes, they can take on features of a cell called the macrophage cell, or a myofibroblast cell, or even like a stem cell, which is shown here. So we took cells from the aorta of our mouse model and grew them in culture, and just asked, did they take on features of these other cells, atherosclerosis cells at baseline? And down here is the cells from the control mice, and the cells in the green are the cells from the mutant mouse. And here's a marker for stem cells. So you can, and what's shown down here is exposing the cells to 3 different levels of cholesterol, because we determined that cholesterol could drive these changes in smooth muscle cells. But here we're asking, do the mutant cells actually turn on all these changes that for plaque formation, even with no cholesterol around? So here is the wild type cells. They're not expressing much of this marker of a stem cell. And here's our arginine, and don't do so even with cholesterol. So this is putting increasing amounts of cholesterol in the cells. But here's the mutant cells, and they've already got this marker turned on. And when we looked at the myofibroblast marker, once again, here's at baseline for the wild type cells in gray, and you can see that it'll go up with high levels of cholesterol in the wild type cells, but it's way high already in our mutant cells. The same for this marker. Plaques often have calcium in them, and it's because the smooth muscle cells start taking on features of bone cells. And here's one of those features of bone cells, and you can see that it's not turned on at 0. And in the wild types, it's not turned down at 2.5 mg per ml cholesterol outside the cell. But with that low level of cholesterol, it gets way upregulated in the mutant cells. So either at baseline or low levels of cholesterol, our mutant cells are starting to take on these features of smooth muscle cells that are found in plaques. And for the wild type cells, they either didn't take them on, or they took them on at very high levels of cholesterol, this 10 micrograms per ml. So here we have a cell that's more prone to form atherosclerotic type cells. And we wanted to figure out why this was. And I'm not going to go through all the data, but we went back to the fact that the cells, this arginine 149C, the actin was going into that complex and getting stuck. And what happens typically when something gets stuck in that, the cell becomes stressed and they start activating factors to correct the problem. And one of the factors they activate is called heat shock factor 1, or HSF1. And this is a cell stress protein that gets turned on from a bunch of different factors that can activate cell stress, including, it was first described by just when you just heat up cells above 30, or typical temperature that our body is, and just heat them up a little bit, just a few degrees, they turn on this HSF1. And now we're finding this mutant actin is turning on HSF1. But then what we found was that HSF1 was activating specifically cholesterol biosynthesis and upregulating a gene called HMG-CoA reductase. And with that, there was an increase in the amount of cholesterol in the cell. The cell was suddenly making more cholesterol because HSF1 was turned on. And that was driving increased cellular levels of cholesterol and turning on those modulation markers. So here we have the wild type cells where you had to put cholesterol on the cells to turn them on. But in our mutant cells, this pathway was turning on those modulation markers because it was increasing the amount of cholesterol the cell itself was making. Now, the beauty of this pathway is the turning on these enzymes like HMG-CoA reductase is responsible for the increased cholesterol in the cell. And we can block that pathway with statins. If you come in at statins actually triggers blocks HMG-CoA reductase, and that's why it's so effective in blocking cholesterol made by the liver. But it turns out it can also block cholesterol made in smooth muscle cells. And we went on to show that if we took on mutant mice with this the ACTA2 arginine 149C mice on that ApoE background. And here's our just ApoE, our control mice, and the amount of atherosclerosis, once again, is all these red streaks in here. You can see the increased amount in our ACTA2 compared with our control. And both of them go down to some extent, but you can see that with statin treatment, and in this case, we use pravastatin, but any statin would work. It's just that the mice tolerate pravastatin the best. And with that treatment, you can see it blocks almost all the atherosclerosis in this mutant mice. And that's quantified over here. It definitely boxes it down to what is there in the wild type. And this was without altering the serum cholesterol at all. I mean, it's pretty amazing. And this really tells you that that how much cholesterol you're making inside the cell can also drive atherosclerosis, not just how much cholesterol there is outside the cell and coming into the cell. So right now, this tells us that we can block the increased atherosclerosis associated with some of those ACTA2 variants that predispose to atherosclerosis by just with the statin. And we were recommending that individuals with ACTA2 talk to their doctor about their risk. And we're trying to figure out which variants increase risk and which don't. And I'll talk about that in just a minute. And then if you do have an increased risk, that you go ahead and start the statin, have a calcium score in your late 20s, at the latest in your early 30s. And if the calcium score shows any evidence of atherosclerosis, go ahead and start a statin at that point. And we more have, as we get more data on that the calcium scores on patients with ACTA2, we may end up altering that recommendation. But right now we don't have much data on calcium scores with ACTA2 patients. But we know that is a good marker of atherosclerosis already if you come up with a calcium score above baseline, that you're already starting to form those plaques. And like I said, those are forming because smooth muscle cells are turning into that bone-like cell, and they're doing that also in our mutant cells. So I'm not going to go through a lot of the details. So are there any questions about the atherosclerosis? Are there any questions in the chat? Did Ellen and Chris-Ann, did you follow that?
No, that was super interesting. I feel like I've never understood it so well. So this is having heard much of this internally presented before, this is very helpful for me. There was one question, which I think you were talking about here at the end, but they asked, in ACTA2 coronary artery disease, is there significant calcification in individuals with a premature CAD presentation? When should ACTA2 be suspected in an individual with premature CAD?
Yeah, I think a calcium score will be good to assess plaque burden in patients with ACTA2 arginine-149C. The plaque looks just like it does in the wild type mass. Unfortunately, we don't have many plaques to look at in patients with ACTA2 mutations. And that's because, with any sort of surgery or stenting, we don't get the tissue to actually look at it. But the cells themselves, even the cells from humans, and I haven't gone into that, they tend to modulate into the bone-like cells, more rapidly than the wild type. So I still think calcium will be a good marker for the extent of plaque. And those calcium scores are performing very well in predicting who needs to get on statins in the general population. So I think it'll work as well for ACTA2 arginine-149C based on our data, but stay tuned. We continue to collect information about the atherosclerosis and about those calcium scores and the extent of atherosclerosis. And I talked a little bit about the extent of atherosclerosis, that in patients with ACTA2 arginine-149C, that it can extend to other arteries in the body beyond the coronary arteries too. So we need to look elsewhere.
She also asked when should it be suspected in someone with early onset CAD. So I guess
I think if the specific variants is going to be early onset, and we know about these two variants here, arginine-118 and arginine-149C. This is actually the most common variant we see in patients with ACTA2, this 149C, and it's about worldwide, about 25% of the people with an ACTA2 variant will have this arginine-149C. So it's very common. And what we can do is start assaying for this pathway. What we're doing now is assaying every single variant in the gene, because now we know the variants that don't cause atherosclerosis don't activate HSF1, and those that do cause atherosclerosis will specifically activate HSF1. So Mauro and Shuyan from my lab are busy. Mara is collecting all the clinical data, and many of you got surveys to see if you have any evidence of atherosclerosis, and thank you for filling out those surveys, because we want to match up that data with the mutations, our assays of the mutations, to make sure that this aligns 100%. And then as we identify new variants, we can quickly assay them to see whether they increase HSF1, do they drive HMG-CoA reductase and increase cholesterol in the cells, and so on. So we'll have the first set of assays and the variants and the correlation. We'll have that hopefully published by midsummer or late summer. So be happy to do another seminar just on ACTA2 mutations and how we set up this assay. So other questions?
Okay.
And so far.
Okay.
It's just the one, yes.
Okay.
I think you're doing beautifully with explaining it in
So.
detail.
It's really complicated, and it only gets more complicated. I made this a little bit more simplistic than that last one. So we now know that the pathway to CAD, but what is the pathway to moyamoya disease? And we've been working on this for a long time. We first found the smooth muscle dysfunction syndrome, and then with Ellen Regalado sort of leading this effort, we really defined all the complications of the syndrome and made recommendations for the management. So we want to continue our research and really build on these management recommendations, along with other groups that are working on this too. And we know it's that specific change, arginine 149C, and this protein is only almost exclusively expressed in smooth muscle cells. So we come back again to those smooth muscle cells for this. And we've known about the 149C, but recently, the 179C for smooth muscle dysfunction syndrome. But recently we have had patients referred to us from around the world that had overlapping features with some smooth muscle dysfunction syndrome. So we went through and collected all the clinical data, and Anita Ka, one of the MD PhD students in my laboratory, went through and collected that and really outlined the spectrum of disease, with the smooth muscle dysfunction syndrome sitting on the most serious end of the spectrum, and then going through to the 258 and all the way to an arginine 39H. So, and with each of these, here's smooth muscle dysfunction syndrome with the PDAs and the eye problems, the moyamoya-like disease, the early onset thoracic disease, pulmonary disease, and problems with the bladder and the gut. And then you can see that there's overlap with some of these features with the other mutations, but none of these other conditions has all the complications. But this is telling us something about the molecular biology. And that's one of the reasons we wanted this kind of detailed map in terms of what variants are causing what. And then we know that the serious problems in a smooth muscle dysfunction syndrome are the cerebral vascular disease, because that can lead to strokes, and there's just not a good option to treat this. The aortic aneurysms and that lead to dissection, there's a great option for treating that. It's not a fun option, opening up cardiovascular surgery to go in and repair the aorta. But once you put that graft in, it's a man-made material and it lasts extremely well. And people have had those grafts in for 50, 60, 70 years. So we really, it's not a great treatment, but we know how, if we know somebody has this condition predisposed, we can prevent the dissection deaths. And the thing we understand least is the problems with the arteries, and we'll be working on that next and working with some experts in pulmonary disease. But what I'm going to talk about is our work on moyamoya disease and trying to understand, like globally, why this particular variant, that 179, is so much more severe than all the other variants. And so we start out getting cells from patients, and then we can differentiate those into stem cells that can be any type of cells, and then take them into smooth muscle cells. We made an SMDS mouse model where we knocked the mutation in to just smooth muscle cells. And then we can take smooth muscle cells from this mouse model and from the patients themselves and grow smooth muscle cells in cultures. So we've used these three different systems to really understand what's going wrong with this variant. And Callie Kortler's work has really been focused on what is the basic defect with this mutation. And it's a whole nother talk about what goes wrong, but we found out that smooth muscle alpha actin actually moves into the nucleus and has a nuclear function. And the nucleus is the command center for cells that tells cells how to do everything. And that's probably because this arginine 179 disrupts the ability of the actin to move into the nucleus. That is probably why it is the most severe mutation. And we don't know why this disrupts movement into the nucleus. We have some good data that may explain it, but we have to work a little bit harder. But what we know happens when the smooth muscle alpha actin that's made by the ACTA2 gene, when it doesn't go into the nucleus, we know that the cells do not become fully differentiated smooth muscle cells. So in our body, as we develop, we have stem cells. In the case of the cells and the arteries that go in the ascending aorta in the brain, those are made from neural crest stem cells. And in the case, the arginine 179 doesn't allow the neural crest cells to completely differentiate into a smooth muscle cell. And because of that, they end up with an altered smooth muscle cells in their arteries, in their gut, in their eyes, and so on. And that's what's causing the problem. So one of the issues, though, that is very clear, or was very clear early on when we had we're looking at the cells in culture, is that the control smooth muscle cells, if we put them in a chamber where they have to move from one place to the other, that over time just a few cells move. But when you look at the SMDS cells, they move like crazy. They are running like crazy. So already this sort of suggests maybe that increase in migration is one of the reasons why the arteries in the brain get occluded in this condition. So, and if you look at the arteries themselves, so here's the lumen inside the artery should be completely free of cells. There should be no cells inside, and the smooth muscle cells should be staying in the wall of the artery. But if you looked at one of those blocked arteries in people with moyamoya disease, here's the smooth muscle cells, which are these purple longish cells here. Those are the ones in the wall, but here's where it's supposed to be completely open for blood flow. And you see there's all sorts of cells here, and they stand positive for smooth muscle cells. And you don't see the cholesterol at all. There's no cholesterol in there, so it's not atherosclerosis. And you don't see any inflammatory cells. You don't see the calcium. You don't see those macrophage cells, those inflammatory cells. So what you've got is just a lot of smooth muscle positive cells inside the lumen. And so we took our SMDS mouse model and wanted to see at baseline, when we imaged the arteries in the brain, we didn't find too much. The arteries were straightened, and we see that in SMDS patients, that they tend to have straighter arteries in their brains, but we didn't see any blockage. So we decided to go in and do a injury to the left carotid in these mice. And that's just tying off the left carotid. And that means that there's a clot that forms throughout the carotid artery. And over time, so we've blocked the artery, there's a clot that forms, but over time that clot goes away and you may be left with a few extra cells inside the lumen, but most of the smooth muscle cells will be in the wall in a control mouse. So we wanted to see what would happen with an SMDS mouse. So we did the same injury to that left carotid artery, and here's the artery in the SMDS mouse. And you can see inside the purple cells, there's nothing in the lumen left over. The clot's completely gone. And you can see that the pink cells here are those smooth muscle cells, and they are all in the wall. But here's the SMDS mouse, and you can see right away the artery is bigger. And here's the wall. It's very, very thinned. and the inside of the artery is completely filled with cells. And once again, it looks a lot like what we see in patients, these types of lesions. So we have now an SMDS mouse model that forms a lesion in the carotid artery that looks like a moyamoya disease lesion. And we've gone through and quantitated this. And also with that injury, I didn't mention this, but with that injury, 20% of the SMDS mice die of strokes. And we can see that they're having strokes before they die. And so, and none of the wild type mice die of strokes. So not only do we have the lesions that look like the moyamoya disease problems with the artery, but the mice, a small number of mice are actually dying of strokes when we do this to the SMDS mouse. So we wanted to ask, is there some way to slow that migration into the lumen? Is there some way we can block this? And we know that stem cells use glucose from the blood for their source of energy. And that if you take a stem cell and make it use a different source, and that's called oxidative phosphorylation, where they switch and start using the energy source called the mitochondria inside the cell, that it actually will differentiate stem cells all by itself. All you do is have to metabolically change the cells from taking up glucose to oxidative phosphorylation. So we went in and tried to see if that would help differentiate. And we first checked to see if our mutant cells were using glycolysis. And this is just a graph of the amount of glucose. It's actually you're looking at the amount of glucose metabolized to lactic acid that's being released by the cell. But the smooth muscle cells are taking up a lot, the mutant cells, those ACTA2 arginine 179 or SMDS mutant cells are taking up a lot more glucose than the wild type cells. So we've confirmed they've even had this metabolic, they've stayed like a stem cell in terms of their metabolic program. So we wanted to see if we could give the cells a drug that pushed differentiation. And this is just, we gave them a drug called nicotinamide riboside. And that's a drug that drives it provides the precursor for oxidative phosphorylation. So it makes it much easier for the cells just to start using oxidative phosphorylation and turning off glycolysis. And with that, they differentiate better. I won't go through this plot, but over here, I want to show that it affects migration. So here's the wild type cells in gray again. And this is the wild type cells without the nicotinamide riboside, the NR, and treated with NR. So even the wild type cells don't migrate as much with NR. But here's the mutant cells. So first of all, you got a lot, they're running around like crazy. But if we treat them with NR, they actually come to the level to the untreated wild type cells. So we effectively prevent that migration. And so we wanted to see if that would work in our mouse model in preventing, number one, the strokes, but also those lesions in the carotid artery. So here is the days after ligation, and the wild type with or with NR is in black and red here. So this is a survival curve here. And all of our wild type mice, as we go in and enter the left carotid artery, they survive just fine. And here's our untreated SMDS mice, that 20%, 25% that die of strokes. And here's, if we treat them with NR at the time of the ligation, the number of mice that are dying of stroke is less. And to make all this statistically significant, we'd have to do hundreds of mice. So we're probably not going to pursue that. But this is very encouraging that it's preventing ischemic strokes in the SMDS mice. And then we looked at the lesions, and that's shown over here. Here's the wild type in the SMDS mice treated with just a vehicle or just with the stuff we put the NR into, which is just water. So they're not treated with anything. Here's those lesions. And if we give them NR, here's the wild type again. And now here's the mutant mouse. They're with little to no cells in the lumen. The lumen is clear and open. Now the artery is still bigger than it is in the wild type. So if we gave this the sort of enlargement that SMDS patients already have in their carotid arteries will remain there. It's not going to change that. But we've blocked this lesion. And this is all quantitated over here. Here's our wild type mouse. And then this is the amount occluded in the mutant and what happens with NR. And this is just showing that the lumen, which is the inside of the vessel in both of these, that it goes up and stays big in the mutant with the treatment, but it's wide open. So we think this may be a good treatment for preventing moyamoya disease. And our next step is to go ahead and let these mice form these lesions and then treat them with NR and see if it makes the lesions go away faster. And so stay tuned for that, because we know many kids have the some occlusion of their arteries in their brain at the time of diagnosis. And we've also done imaging in the mouse, and this is probably here in Houston is the best place in the world to get this imaging done. I don't think there's any place else that can do this level of detail. We wanted to see, was it actually, so we're just looking at the artery here in the mouse, and we wanted to see what was going on with the arteries in the brain with the NR treatment. And I'm just going to ask you to focus in on this artery here, which is in the SMDS, and you can see it's pretty narrow. But with the NR treatment, it actually opens up, and we're just starting to, this is a diameter of the wild type, and then the SMDS mouse, and then this is the SMDS mouse with the treatment. So we can see that some of the arteries are actually opening up, and we've done a lot of studies looking at collateral blood vessel formation. And we see a lot more collaterals forming because they have to with the mutant mouse, and all that goes back down. So we're now going in and sectioning through these arteries to really document that there's changes in the arteries in these areas. So we hope to have the nicotinamide riboside, use it in a treatment trial. I want to mention that there's other treatments that have the same effect on the smooth muscle cell that we can go on to try or do in combination with the NR in the future. So we're trying to work through that, other therapeutics. And to be honest with you, if we just take away glucose and force the cells to use the oxidative phosphorylation, it does even better. It changes the cells in the same way that the NR does. And that would be equivalent of putting a child on a low carb diet, an extremely low carb diet. But they do that already for conditions like uncontrolled seizures and so on. So they are able to do that in young children. So we want to see if other therapeutics will work. We want to make sure that this kind of treatment, it doesn't, we don't want to make the moyamoya disease better, but make the aortic disease worse. So we now have SMDS mice on long-term treatment with this drug just to make sure it doesn't make the aortic disease worse. And we're about a little over a half a year into that treatment, and there's no evidence it makes the aortic disease worse. We've applied to the FDA and the Federal Drug Administration and our institutional review boards for approval to start a study of NR in patients with MSD and SMDS, and FDA approval was received in record time. So we're still waiting on the ARB. And we're going to start out by just figuring out imaging protocols and data from blood collections to really confirm with NR that we can change the phenotype in human smooth muscle cells. And we have imaging and blood studies that we can do to prove that. And then we'll use that data to actually design, to start the clinical trial. So the IRB, we're working on the final changes, but we hope to start bringing people to Houston in June or July at the latest to start this part of the study. And so we filled in this arm, and we've got a treatment for at least the moyamoya disease. Like I said, we're working through a large amount of funding from a foundation called the Leduc Foundation to really understand those triggers of aortic dissection. But in the meantime, we have a good treatment for that in SMDS patients and in ACTA2 patients in general. And then now we have a treatment for the CAD, just that high, pretty rigorous treatment with statins. And I just want to thank everybody in the lab. These are the people, Callie Courtney, Abhi Chattopadhyay, Anita Kala, and Kavita Kav, MD PhD students who are sisters who are in my lab. Pooja and Gwen, an amazing graduate student that does all our bioinformatics. And then Suvivi Majender. And then Mara Barrio is the one that's sending out those surveys and collecting all the data on atherosclerosis in patients with ACTA2. And then a lot of other, and really appreciate the John Ritter Foundation, GADA for funding, the Leduc, American Heart Association gave me their biggest honor as a grant to study SMDS, and that's a merit award. That is the highest prize they give, and I'm really grateful to them. And then we've had a large number of families, the Olivia Petra Cohen Research Fund and the Temerity Foundation, and some other families that have donated money for these studies. So thank you. And I have time to answer just a few questions before I'm going to have to run.
Yes, thanks, Dr. Milewicz. This was very informative and I think very nicely explained. There's one question here in the chat box that's asking, is it possible for people to have more than one variant, ACTA2 variant? Can a patient?
It does happen, and we have seen that in 2 patients out of the 500 or 600 ACTA2 patients we have in our cohort. So it's extremely rare.
That I just have to say, this is so exciting to see, having,
moving forward.
Yes, for many years we've been talking about this, and it's really great to see the FDA approved it. It's amazing, a big step and a big hurdle. So congratulations to you and everyone who has been helping you in your lab with this work. We did have a question specifically about the treatment, which says, is it possible that the NR treatment may also help other symptoms of SMDS?
Oh yeah,
bladder, etc.
I didn't have time to talk about that. I didn't think it would help like the bladder and the gut, but we had a surprising finding. So ACTA2 is really responsible for contraction in the arteries, but there's a different alpha actin that is responsible for the gut. And so I couldn't quite figure out why, the one that was the mutation in the gene that made the actin for the contraction in the arteries was disrupting the gut. But it turns out when that smooth muscle alpha actin must move into, when it moves into the nucleus, it's also important for turning on that gut actin, which is called gamma actin. And that same gamma actin is responsible for the gut and the bladder contracting, because the smooth muscle cells are contracting like this every pulse pressure, whereas the gut and the bladder contract like this, and they let go, and then they correct. So it's a different kind of like biomechanical mechanism. But it turns out, so the smooth muscle alpha actin, if it's not in the nucleus, it turns off gamma actin. But when we give NR, it turns it back on. So amazing. And there's nothing wrong with the gamma actin. It's just that the alpha actin is disrupting it, because it's not going in the nucleus, isn't allowing it to turn on. So it must be regulated by alpha actin, but once it gets turned on, it should function just fine. So we'll see. I think it'll work very well for the gut and the bladder, but we'll have to see. I'm optimistic. I was surprised by that, really.
Yeah, that's amazing.
That's one of the fun things about research. It always surprises you.
Well, I'll just also add that there are people saying thank you in the chat, and I understand much better. Thank you. So I think people got it successfully. There's not any questions, but I think it's kind of washed over people, and it's really exciting.
And I want to say that I can't make it to the ACTA2, the SMDS meeting next week. I have to be in Amsterdam for a Leduc meeting because I'm leading this research program. I have to be there. But David Murdoch, Dr. David Murdoch, a geneticist that's in my group, will be there to present this data and probably a little bit more detail than I'm able to present it here. A lot more detail into the other treatments we're trying also to be either an alternative so that people have a choice of what to do, or in combination if it works better that way. So he'll have a lot more data to present and go through it again. If you didn't get it all this time, you can, I think you can join that through webinar. Yeah. And I think he's the first speaker there.
Great.
Yeah, Dr. Milewicz, I know you're in a rush to go, but there is one last question here. I know you presented on 149 and 179, but there's a question here. Is there a recommendation for testing for cerebral vascular disease with positive ACTA258?
I think that we would recommend some baseline imaging for moyamoya disease in that condition, because sometimes the first presentation is a stroke. But what age and how frequently, we really, we don't know that. I think right now I would recommend, if it was my child, I'd go ahead and get imaging as soon as the child was old enough to sit still and not have to be anesthetized during the imaging, and then have it rechecked every 5 years. So, yeah. Right now, we think that more than half the patients with arginine 258 will have some degree of the moyamoya occlusion. And what we're finding in the 179 should apply to the 258 in terms of treatment, because that also disrupts the 258 prevents the smooth muscle alpha actin from going into the nucleus also.
Thank you. Thank you so much.
I just wanted to also say that this was recorded, so we will make it available. So watch your inbox, and just add our ways to contact us on social media and website and email and all of that. But thank you, Dr. Milewicz, and thank you,
Chrisanne. Yeah, thank you, guys.
Yes, thank you. Thank you, Dr. Milewicz.
Yeah, this is super informative.
Yes, and thanks to everyone who attended again. It's just always good to know the interest in research. Thank you, Dr. Milewicz. I wish you the best too. Congratulations on all your research.
Oh, thank you, Chrisanne. Thank you, Ellen.
Thank you, everyone. Bye-bye.